Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson, your host from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries. Today, we're rebroadcasting an episode featuring Leakey Foundation grantee, Christopher Schmitt. He discusses vervet monkey life histories and much more. We hope you'll enjoy this broadcast, and if you're watching live, please be sure to add comments to the chat, as I'll be watching the episode along with you. We also have a special exclusive series of events starting next week, the Summer Travel Series. This series virtually whisks you away to research sites around the world, and the proceeds from the series go to the Leakey Foundation's Baldwin Fellowship Program. We've shared a link to learn more or register in the chat, and I hope we'll see you all there. Now, let's begin Chris's episode.
Here with us today is Leakey Foundation grant recipient Christopher Schmitt. Chris, thank you so much for being here with us.
Well, thanks so much for having me.
Chris is joining us from Boston, where he is an assistant professor of biological anthropology and co-director of the Sensory Morphology and Anthropological Genomics Lab at Boston University. His research has taken him all over Peru to study yellow-tailed woolly monkeys wherever they are found, which there's quite a few locations in Peru where they are found. So also to the Suttering Nature Reserve and a private farm near the Harip Dam in South Africa to study vervet monkeys. They're really cute and a gorgeous, gorgeous location. And also to the Lilongwe Wildlife Trust in Malawi, also to study vervet monkeys. Again, just like gorgeous. I don't know. I can never, I feel like all of these field sites we get to see are like, I couldn't pick one. They're just all gorgeous.
Yeah, it's amazing.
Yeah. So today he discusses his work using morphology, behavior, and genetic techniques to understand primate growth and development. But before we hear from Chris, if you are watching us live on YouTube, Facebook, Twitter, or Leakey Foundation Live, you can post questions for Chris at any time during the chat, and he'll answer your questions live during the episode. The earlier you get those questions in, the more likely he will be able to answer them. So again, get those in early. My first question is a question about questions. So what are the questions that you are working to answer through your research?
That's a good question. So I'm really interested in growth and development. So this has kind of been a consistent interest throughout my academic career. And I started out really asking questions about behavioral development. So my PhD work done at the Tipitini Biodiversity Station in Ecuador at New York University with Tony DiFiore was looking at behavioral differences during development between woolly monkeys and spider monkeys. From there, I got a postdoc at UCLA, working with the International Vervet Research Consortium, really trying to understand, rather than behavioral development, looking at morphological development and growth, and doing this both in captivity, where we can construct really good growth models, but also looking at it genetically. So really trying to understand how these growth patterns are controlled for in the genome. And so with that research under our belt, like in captivity, I'm also kind of taking it back to the field to better understand the ways in which we can kind of utilize this genomic resource and what we've learned in captivity to understand the evolution of these growth patterns in wild populations, with an eye towards better understanding in particular how growth patterns related to obesity might actually be organized through evolutionary processes, and using vervet monkeys as a model to understand that.
The trajectory of your career and your research has, at least to me, seems like it's unfolded and incredibly holistically. Tell us about when you became interested in studying animal behavior and how that led to studying monkey behavior.
Well, it definitely feels a lot more chaotic than holistic when you're kind of going through it. But yeah, I've tried to kind of keep this main through line of understanding growth and development like key throughout my research. But I really got interested in behavior to begin with, because when I was a kid, I was really awkward around people. I got along much better with animals, like pets. And this is me with my cat Odetta, just an example of how much I love my animals. And so that really kind of led to a really great opportunity I got through my high school. I went to an inner city public school, Milwaukee Public Schools, and we were very fortunate to have a benefactor who had graduated from the high school who gave a fund to an organization called the Earthwatch Institute, which is still active today. And through the Earthwatch Institute, I got this opportunity to work with wild black and gold howler monkeys in Argentina on a project run by Dr. Govindasamy Agoramurthy. And so I went there for a month, studied howler monkeys, and usually, no offense to people who study howler monkeys, they can be kind of boring. They sit there, they poop, they howl. But in this case, it was really amazing. The monkeys had intergroup encounters, they fought, they dispersed, like I saw copulations. It was like the maximal howler monkey experience, at least in my experience of howler monkeys. So I, got really, really interested in it. And when I went to college, I went to University of Wisconsin at Madison, where I majored in zoology, thinking that was where monkey work was done. I didn't really understand at that point that most of that work in the US is done in anthropology departments. And I had a double major in English literature and a certificate in Chicano studies, because why not? And while I was there, it wasn't until I took a primate behavioral ecology class with Dr. Karen Stryer, who became my undergraduate mentor, that I really kind of got on this path towards being a biological anthropologist studying primates. And again, I kind of started out with primate behavior. And after I graduated, not really knowing what I wanted to do yet, I essentially applied for this year-long field assistantship working at Susan Perry's site at Lomas Barbudal, working with white-faced capuchins. And from there, I was just completely sold. I was like, this is what I want to do. The capuchins were amazing. Her research project was really focused on development, and that was really kind of pivotal. That kind of steered me for the rest of my career, being interested in behavior and growth and development. And so, yeah, that's kind of how it all started out.
Do you want to talk a little bit more about your field experiences in with the capuchins and some of your other early?
Yeah. I mean, Lomas was like hardcore field work. They were like, it's in a wasp and bee sanctuary, so like getting stung all the time. The capuchins were really, really difficult to follow, but also really personable and amazing. It was like watching a soap opera every day. And on top of that, Susan Perry's field site has been going since, the mid-90s. And so when I joined it in, the mid-aughts, there was already this really rich kind of database of behaviors, and we knew who everybody was related to, more or less, at least through the pedigrees that were established from watching individuals give birth and interact with each other. And so it was this amazingly dense data resource that when you jump into it is just incredible to have all of that knowledge and jump into it and be contributing to this large field site. And it's really a shame, because I keep hearing that these long-term field sites are having a lot of trouble maintaining themselves. But I hear that the Leakey Foundation is actually kind of working to address that at the moment.
Yes. So the Leakey Foundation understands the incredible value of long-term research. So we, especially for those projects that are facing gaps in funding or unexpected emergencies. So the Leakey Foundation created a lifeline for these projects called the Primate Research Fund. We've shared a link in the chat to learn more about the fund, and please check it out if your site is experiencing these kind of circumstances. If you're interested in helping to support the Primate Research Fund, we've shared a couple links too with ways you can help.
No, that's so amazing. Thank you for doing that. Like the idea of those capuchins no longer being followed, it's horrifying.
I know it is. It absolutely is. And during COVID, I mean, it's really, I mean, primate sites are hurting.
Yeah, it's so important to keep them going.
Yeah. So you also work with howler monkeys.
Well, that was only my first field experience, but in my dissertation research, I did work with some closely related species. So yeah, my dissertation work was on primarily woolly monkeys, but also a bit on spider monkeys. So again, at Tipitini Biodiversity Station in Ecuador, so this is an Amazonian field site, following them around from sunrise to sunset. And again, I was really interested in looking at differences in behavioral development between these two species, because even though they're very closely related, woolly monkeys tend to take a lot longer to reach reproductive maturity, almost twice as long as spider monkeys. And so I was really interested in understanding why that was, looking at behavior. So trying to understand whether there's differences in food competition, differences in predation regimes that might kind of organize these very different life histories, even though they're very closely related. So yeah, it was a really great experience, like months and months of fieldwork. I think I did 24 months of fieldwork overall on those species. So really, really amazing experience.
So your work now focuses mainly on vervet monkeys. How did working with them come about, and why are they a good species to address the kind of questions that you're looking at?
Yeah, so after I got my PhD, well, actually, while I was writing up my PhD, I saw a job ad on the Primate InfoNet, which I think is still active. They have a job board run through the University of Wisconsin that shows you, different career levels of jobs that you can get working with primates. And I saw like a kind of like cryptic ad saying, we're looking for somebody to go to Africa and trap hundreds of vervet monkeys. Call if you want more information. And I was like, okay, this sounds crazy. And one of the people on the ad, Trudy Turner at the University of Wisconsin-Milwaukee, who's now one of my main wonderful mentors in my career, Trudy, I called her because I had met her recently at an NSF function. And, I'd had experience darting monkeys during my dissertation to put radio collars on. And so, the transition to trapping was kind of very easy. And so I ended up getting the job. And a few weeks after defending my dissertation, moved to South Africa in order to begin working with the International Vervet Research Consortium. And the original idea of this consortium was to really better understand SIV in order to better understand HIV in humans. So these are immunodeficiency viruses that the vervets have, but the vervets don't progress to AIDS. So we really wanted to understand physiologically why that was. And in that process, we trapped hundreds of vervet monkeys around Africa and the Caribbean, where they also live, and characterized a lot about them phenotypically. We got hormone information, we got immune information, we got morphological information, and really building up this massive resource. And I've been working with it ever since.
Awesome.
Yeah.
So your field site is a little bit different than other field sites, or at least your field sites in South Africa are a little bit different than other field sites we've heard about before on Lunch Break Science. Could you tell us a little bit about your site?
Yeah. So unlike most of the sites where I've worked in my career, like Susan's site, like Tipitini, I don't have a stable field site that I'm kind of working to maintain. Since we were going around trapping a lot of monkeys in a lot of different locations, when I started my faculty position at BU, I just thought, okay, well, which of those sites were the most productive and which do I want to go back to? So Siptering Nature Reserve, which is where the photos that are flashing on the screen right now are from, was one of those sites. We had a lot of luck getting a lot of samples from monkeys. And it was really a nice place to work. We had these chalets we were working at, really good field site. So I decided to go there as a site that is kind of undisturbed, relatively speaking. And then Harib Dam, which is on the screen right now, this is a site only about 200 kilometers away from Siptering. Very similar ecotypes, but instead of being in a nature reserve, it's on a farm, like an active working farm that raises horses. And so the monkeys there are exposed to a lot of anthropogenic impacts. They sometimes get electrocuted by power lines. They get shot at by farmers. And they also have access to a lot more human-like food. So they steal the food of the horses. And for me, this seemed like a really good model because rather than having this trouble of constantly having to get funding to keep the research site going, at the farm and at Siptering, they already have infrastructure in place that really allows me to just come and go. And then when I'm there, I can provide funding, I can provide work, we're interacting with local people. And when I'm gone, it's still being maintained because one is a working farm and the other one is obviously a nature reserve that's being run by the state.
Tell us a little bit more about the team that works with you at the sites.
Yeah, so usually it's a combination of local folks and students that I bring from BU. So I have a veterinarian that I work with locally, and that's required. Obviously, when we're trapping monkeys, we want them to be healthy and happy at the end of it. So he really kind of safeguards the health of the animals. And then the students that I bring from BU, I really want them to get kind of a whole experience of the field site. So when we get to South Africa, a lot of times they have never heard of apartheid. They've never heard of the history of the country. So I make it a point to take them to places like Pretoria, to Maropeng, to kind of get the human evolution history of South Africa, also to the apartheid museum so they can really get that social history of the country. And then also, of course, when we're at the field sites, we're working with local folks in the park, we're working with the farmers. So yeah, I really want them to get a sense of what it's like to work in country instead of just kind of coming in and taking what we need and going again.
Well, I'm sure that they get so much more out of it that way.
I hope so.
So you've also worked to raise awareness about being queer in the field through your blog, Things I Learned as a Field Biologist. You also help run the Queer Bioanth group for the American Association of Physical Anthropologists and are featured on the site 500 Queer Scientists, all of which we have shared links to in the chat. What is it like to be queer in the field, and what can we do to help better support our fellow queer scientists?
Well, I'm really lucky being a biological anthropologist because I feel like our field has been doing a really, really good job being supportive of queer students, queer faculty members. But having said that, like we're not always in those professional contexts, like at a conference or in our departments. And so the field is one of those places where being queer can be very difficult, and sometimes there are problematic aspects of that in relation to the national context, the local context, and all of that, of course, is intersectional, right? So, me being a white guy from the United States, even though I'm queer, like those identities will buffer me from some of the potentially more dire consequences of being queer in the field. But even in those cases, like, for example, I worked for a bit in the Gambia, and at the time that I was working there, the president, Yahya Jammeh, was talking about slitting the throats of all the gays. And so it was really important to like keep closeted, but it was also like very stressful. Like my boyfriend was breaking up with me at the time, and I was like, hi, I can't talk to anybody about this. So, it can be very difficult even when certain intersections of your identity do kind of buffer you from those effects. But again, if I were more femme presenting or female presenting, I would face a whole slew of differences that intersect with my queerness that make fieldwork, more difficult still. And, that's not to mention, like, if I were a person of color or a local person who also was queer, very, very different problems that might be faced within those contexts. So the main thing that I do, when I give presentations at universities, I always try to talk about this as well. The main thing that PIs can do to kind of help with that is just, really be conscientious about the fact that there are queer people who are in our field who, maybe don't want to be closeted in their professional lives. And, some of us have to stay closeted, and there's nothing wrong with that whatsoever. We have to do what we need to stay safe, but that also can have repercussions. So, so really what I ask PIs to do is follow the same kind of protocols that they would with any other kinds of marginalized identity, set up really clear, good codes of conduct that address these identities at your field site. On top of that, have consequences for when people violate that code of conduct, right? If somebody says something homophobic and you're like, oh, they don't usually do that, it's like, that's not helping that queer person at your field site to feel like this is a place where they belong, right? And then on top of that too, have really clear guidelines for how to address when things like this happen. With those factors, what the SAFE study shows us, what various other studies that have looked at gender-based harassment at field sites show us, is that having these in place can really help marginalized identity, people who have those marginalized identities, feel safer at those field sites and be more productive, because then we don't have to spend our whole time when we're, should be working and thinking about the work, worrying about like, oh, was my list too limp? Like, was I lisping? Like, did I give away the pronouns of my boyfriend? Is that a problem? Do I have to worry about that? That kind of cognitive load gets to be a little bit too much when you're in the field. So really, what I encourage PIs to do is really kind of help take that burden away from people with marginalized identities like queer folks, by having these very clear guidelines in place.
Well, thank you so much for at least briefly kind of touching on that. And for those of you watching, please check out the links we shared in the chat. I mean, it could be its own lecture topic, So please, again, just share out the resources. And thank you so much for sharing more about that. And now let us hear more about your research. We'll turn over the virtual floor to you and dive into your work.
Yeah, sure. And I'm just realizing that I actually like closed my talk window after like testing it out. So just give me one moment to pull it up.
Yeah, no worries.
Rookie error, right? Open recent. And now I'm getting the rainbow wheel of death.
Oh no.
Of course. Okay, now I've got it now. So we are good to go.
Okay, there we go. Okay.
There we go. So I just want to share, I'll see you in a moment. Just wanted to share a little bit about kind of the main thrust of my research program at the moment, which is really trying to understand the evolutionary biology of traits that we associate with obesity, particularly taking kind of a developmental perspective. So, I think everybody in the United States at this point is aware that obesity has been increasing over the years, and at the current moment in time, about a third of US adults are obese. And although obesity doesn't always come along with health concerns, in many cases it can. And in those cases, it costs the economy quite a bit of money. So there's a vested interest, at least on the national level, to really kind of understand the biology of obesity much better. And this isn't only affecting humans, we also see this happening with domesticated animals and various other species in labs, et cetera. So historically, we've really understood obesity through biomedical frameworks. This is where a lot of work linking genomics to obesity really occurs. And in these biomedical frameworks, we've been really successful at finding genomic loci associated with obesity. Like famous example is FTO. This is the kind of original obesity gene. But really, it depends on what phenotypes we're using, what genes we find that are associated with this trait. We also, of course, know that it's not 100% genetic. We know that there are behaviors and developmental trajectories that are more likely to lead to obesity. So, for example, what we eat at a certain time period, critical periods when we put on weight, can have an impact on lifelong body size trajectories. We also know from historical circumstances, like this photo from the Dutch hunger winter after World War II, when food supplies were cut off from parts of the Netherlands, folks were getting, about 200 calories a day. And 50 years later, those children who were gestating during that hunger winter have a higher incidence of metabolic disorders. So we know too that what you experience during critical periods can kind of program your body to have a certain kind of metabolism. And then on top of that, we know that expectant mothers, what they eat, what their body size is, can also have some influence on subsequent body weight. So these biomedical frameworks have been great. We really need to do a better job of incorporating them into evolutionary frameworks. The two kind of main ways that we've done this are thrifty genotype and thrifty phenotype hypotheses. So thrifty genotype looking at selection for resistance to famine, leading to the fixation of alleles for fat deposition, and a thrifty phenotype being more like this Dutch hunger winter effect, wherein we maybe are having epigenetic programming due to nutritional stress, leading to better or more efficient fat deposition later in life. And tying all of this together is kind of what my research program is attempting to do. And as mentioned earlier, we're really using this great model that the International Vervet Research Consortium has set up to dig into these complex traits like obesity. So with the International Vervet Research Consortium, as this map shows, we've got a really great resource set up where we've got a captive colony of monkeys at the Vervet Research Colony at Wake Forest University, where they're pedigreed. We've got 700 of them whole genome sequenced. So it's really good for digging into these complex phenotypes and finding a genetic link. We then can go to St. Kitts and Nevis, which is a bottlenecked population where they came over from West Africa around 400 years ago and kind of took root in the Caribbean. And this is a kind of more inbred population, really good for, again, finding genomic loci, but in this case in a more naturalistic environment. And then, of course, we've sampled across their natural range in sub-Saharan Africa to really get at ecological variation that might be associated with these traits evolutionarily. And these are all just spots where we've sampled vervet monkeys. We have morphological data, all of these other phenotypic types of data that we've collected from all of these sites, including CSF, DNA, RNA, fecal hormones, morphometrics, body condition, microbiome. And we've tagged all these individuals with microchips so that when we go back to these sites, we can collect longitudinal data, which is what I've been doing at the two field sites, Sitduring and Harip, that we mentioned earlier. And the main project I'm working on right now, and this is also funded by the Leakey Foundation, thank you, Leakey Foundation, is looking at drought as an ecological stressor within these populations. We know that drought is a really strong selective force. It's lower availability of water, lower food availability, and the food that is available because of the drought is usually lower in quality. So I really wanted to see how this drought interacts with anthropogenic impacts. So in this case, as we mentioned before, Sitduring Nature Reserve is relatively naturalistic environment. The monkeys are mostly doing natural foraging, whereas this farm in the Harip Dam, the monkeys are kind of buffering any nutritional problems that might happen during a drought, for example, with all of this extra food they can get from the horses, from the pecan nuts that are growing there, and from various other crops. And so when we actually look at body condition across these two sites, skin fold thicknesses do appear to be significantly increased in the farm population compared to the nature reserve. And on top of that, on the farm, we see earlier milestones of reproduction being reached. Milestones of reproduction being reached at an earlier age. So we see age at first birth going down. We see the age at which we see evidence of having multiple births going down. And we're also seeing testicular volume increasing at a slightly earlier age on the farm than on the nature reserve, again, suggesting that these animals are being better provisioned nutritionally. Now, what the Leakey Foundation is funding, along with National Geographic, is looking at gene expression differences between the farm and the nature reserve site across drought-impacted time periods. So in 2010, this was the first time we went and sampled these monkeys with the International Vervet Research Consortium. In 2016, I started sampling them again. 2010 was just before the biggest drought in South African history began. 2016, 2017 is when that drought peaked. And I'm hoping to go back with this funding next summer to collect again from these animals in order to see how gene expression changes across these drought conditions and also across these different anthropogenically impacted sites. And the idea is that hopefully when we're digging into these conditions, we'll be able to see gene expression changes in these genes that in captivity we've associated with obesity. And this would be an indirect way of showing that these obesity-related genes perhaps are indeed responding to selection based on drought, which is kind of the core idea of this thrifty genotype hypothesis. And incidentally, in the meantime, while the pandemic has been raging on and we can't go to the field, I have been working with a collaborator, Ellen Quillen, at Wake Forest School of Medicine, to really dig into this in an already established baboon population, Texas Biomed or the Southwest Foundation, looking at growth patterns and associating them with genes, looking at obese versus non-obese growth trajectories. We're finding a lot of really interesting candidate genes that I would also going to look for in these expression-based differences in the wild population. So a lot going on in my research program right now that I'm really, really excited about. And I'm really, really excited too that the Leakey Foundation is giving me an opportunity to share this with you. If you want to learn anything more about this research program or what I'm doing, or if this sounds like something you want to collaborate on, I would love it for you to reach out to me. Oh, Ariel, you're muted right now.
It's one of the dangers of coming back.
Yes.
So, thank you so much, Chris. As you were talking, I was wondering if you if your work has kind of overlapped at all with the pandemic in the type of questions you're asking.
Yeah, for sure. I mean, aside from having to cancel 2 field seasons, which you know is a difficulty that all of us are facing right now. I also, was at home during the pandemic thinking, okay, well, what can I do that might have some kind of impact on the pandemic? And, as I mentioned, we built up this massive kind of amazing resource with all these vervet monkey data, including genomic data. And I thought, okay, well, maybe I can actually dig into the genomic susceptibility of these monkeys to COVID-19. And that led to a paper with a few collaborators, including Christina Berge at Rutgers and the usual suspects in the International Vervet Research Consortium, really kind of looking at whether or not the ACE2 and TMPRSS2 receptor variation in the vervet monkeys would confer susceptibility to the virus. And we're really concerned about this because wherever vervet monkeys are found, they are getting up in people's business. So, stealing food, stealing alcoholic beverages in the Caribbean, digging through garbage. And so we're really worried that there's potential for the monkeys to get COVID-19 and potentially pass it back and forth with humans. We don't have any evidence of that occurring right now. But what we found was that based on these receptor variations in the genome, it does look like the vervet monkeys are as susceptible to getting COVID-19 as humans. And this was before the research actually came out in captivity that shows that, yes, when they are infected with SARS coronavirus 2, they do develop very, very similar COVID-19 compared to humans. Like they get the cytokine storm, they get the upper respiratory infection, they shed virus through respiratory excreta and through feces. So they're actually turning into kind of a go-to model for understanding COVID-19. And what we found that was interesting was that the Sabaeus vervet monkeys, so those from West Africa that we also have in the Caribbean, they actually have a variant in ACE2 that looks like it would actually confer a bit of resistance to infection with COVID-19. So it would lower the kind of ability for the virus to bind to that receptor and infect the monkeys. So, it remains to be seen whether or not that actually confers resistance. Like, we would have to actually test that out. But interestingly, in the captive colony at Wake Forest, about half the monkeys do have this variant that presumably would confer resistance. So hopefully in future, we'll be able to see if this actually plays out as increased resistance to getting sick from the virus.
Well, I've actually shared a link to that paper in the chat, so be sure to check that out. It's a really interesting article.
Thanks.
Now let's take some questions from our audience. If you are watching live and haven't gotten your question in, be sure to get your question in now, so Chris can answer it. Do we? Hold on, let me. We have one. We have our first question. Cheryl asks, if you could answer one question about human evolution using your research, what one question is most interesting to you?
Well, for sure, I think that, I think I made it pretty clear that I'm really interested in growth and development, and I'm very, very interested in obesity. So I'm fascinated with this idea that humans lay down body fat very differently from other primates. I'm really interested in how we can use primate models to better understand this. So at the moment right now, that's kind of my main obsession. I'm really interested in metabolism, laying down body fat, and what we can learn from non-human primates as a model for understanding the human patterns that we see.
We'll take our next question. H asks, are you hopeful that you will not have to cancel a third field season due to the pandemic?
Yes, definitely. I am very hopeful. In South Africa in particular, they've had a lot of trouble kind of getting the vaccine out. There have been a lot of snafus with the government working with vaccine distributors and getting the right vaccines and getting them in people's hands. So I'm hopeful in part because I'm vaccinated. We've been able to get vaccines, but that doesn't really help the people that I work with on the ground there. I'm hopeful, with the Biden administration, purchasing and giving all of these vaccines that they just announced, I think, a week or two ago, to other countries, that this will improve the situation in a lot of other countries that have had trouble getting the vaccine, and that this will lead to a greater ability to kind of ethically go in and continue working in the populations where I work. So yeah, I'm very hopeful that next summer I'll be able to go down and have a field season.
Do you have what are some upcoming projects that you have in the works?
The work that I'm doing at Wake Forest with Ellen Quillen is pretty exciting. So this is captive work where, since they're in a captive colony, we're able to kind of control their diet. And I'm not the one asking them to change their diet, but they do it pretty frequently. So I take advantage of that. And right now we've got a sample, Ellen is working on right now, of muscle biopsies that we're hopefully going to be getting both methylation patterns, so these epigenetic markers that we think are being changed when diet is different, and also gene expression data. And what we're hoping is that we're going to see kind of in real time, we've got 3 different dietary periods, one pre-dietary change, one immediately post-dietary change, and one a couple years after the dietary change. And we really want to see if the genomic level is a place where we can see the impact of these diet changes. And the baboon work that I mentioned in my kind of work talk there, that makes us really hopeful that we will see some really interesting kind of genomic changes that are leading to these larger patterns that we notice in body condition that we associate with this obesity issue. So yeah, I think that work I'm really excited about. On top of that, I'm really, really excited. We just bought Oxford Nanopore Minion for our lab. So this is a portable sequencing machine. And I've been talking with Mrinalini or Minnie Watsa, who works at the San Diego Zoo, who's been implementing this to really do kind of real-time genomic sampling in the field.
Wow.
So I'm really excited for this, not only because, it'll help us, avoid having to get permits to take samples out of country to do this kind of genomic work, but it's really going to allow us to really develop capacity within primate host countries and train people how to do this genomic work themselves. And I think that is one of the most exciting parts about these portable sequencing technologies. And, you know, I say this like I haven't even gotten into the mail yet. So I'm like, this is like very, very new, but I'm really, really excited about its potential.
Oh, wow. It must be like I'm sure you should do like an unboxing.
Yeah, I should. I'll post it on YouTube.
Yeah. Okay, our next question. Let's see here. Brandon asks, what is the most challenging part of doing your field work? What is the most rewarding?
I mean, yeah, that's a really good question, actually. So a challenging part of doing field work. So the field work I do now with the trapping is actually very short term. So I'm only in the field for a shorter period of time. When I was doing behavioral field work, where I'd be in the field for like a year at a stretch or maybe even longer, that became a lot more challenging, in part because, you know, being queer, like having to stay closeted among certain populations of folks, or feeling like I had to stay closeted. You know, sometimes I didn't have to, but I didn't understand that. You know, that really, that can be very, very difficult. And so I think, you know, now I'm in a situation where I'm there for a shorter period of time, but I'm also in a context where like I have queer colleagues on the ground in South Africa. And so that makes the field work a lot easier. With the trapping field work, it's also a little hard because you're kind of, you know, you're coming in, you're not really getting to know the monkeys. You know, we take good care of them, of course, but I'm not really learning them the way that I did when I was doing behavioral work. So that's a little bit of a challenge. It feels a little bit more removed. But for rewards, it's also just really rewarding to interact with the monkeys. You know, our veterinarian, when we trap them, he also like takes care of their health. So it feels like we're making a difference for those monkeys as well. Yeah, like just like digging into the data and really kind of understanding how they work for me is very, very satisfying.
Let's take our next question. Do you, Meredith asks, do you have a fun fact about Vervet monkeys?
Well, I mean, the most fun fact about Vervet monkeys is that they have a bright aquamarine blue scrotum. And it's fascinating. Their scrotum is bright blue, their penis is bright red. And so they sometimes do what's called a red, white, and blue display. I mean, the 4th of July is coming soon, right? So where they kind of get an erection and like, you know, show their bright red penis, bright blue scrotum, and then their white underbelly to others. And part of why it's so fun, I mean, it's fun that, you know, they have a bright blue scrotum, but it's more fun because we don't actually know why it's bright blue. Like a lot of folks have tried to kind of dig into that phenotype and understand, oh, is it evolutionary? You know, is it a response to hormones or stress? And we don't really have an answer yet. And to the point where, you know, there was one researcher, Melissa Gerald, who actually spray painted their scrota to try to understand whether like there's a certain shade of blue that females find more attractive. And what she found was that no, that didn't seem to be the case. So for the Vervet monkeys, I think that's probably the most fun fact, is that that scrotum is very brilliant and we have no idea why.
Let's see. Do we have oh, okay. My last question for you is, do you have any advice for those who are watching who are interested in studying primate behavior? Yeah,
for sure. So one thing I'll say about getting started in primate behavior is that there are a lot of barriers to participating in this field. And one of them is financial. So, you know, one thing that I would just say absolutely to people who are thinking that this is what they want to do is you need to get some experience to know whether or not this is really what you want to do. The field can be very difficult. It can be surprisingly difficult for people, even if you love the monkeys. And so getting that field experience is kind of a critical first step. And this is something that people will look for if you want to go to grad school and actually do this as a career. But of course, that costs money, right? And as somebody who grew up without a lot of money, this was a potential barrier for me too. And the only way that I could afford to actually go and spend a year working in the field at Susan Perry's field site was that I got a scholarship, like $1,000 scholarship that paid for my field clothes, paid for my airfare, and allowed me to not work for an entire year. However, like there are increasingly scholarships that can help cover these kinds of experiences. So For example, the Madeiras Rainforest Conservancy has a scholarship through the American Society of Primatologists to go participate in a primate behavior field course, usually in Nicaragua or Costa Rica. That's been on hold for the pandemic, but I'm hopeful that it'll come back. Field Projects International, which is operated out of Los Amigos Biological Station in Peru, they have scholarships, including a scholarship for future Black scientists, an opportunity scholarship for local scientists in Peru, and a global studies scholarship for folks in the US or Europe or anywhere else around the world to come down and take part in their amazing field schools, which include a genomics in the jungle field school using the technologies that I am so excited about that Minnie Watsa and her colleagues are pioneering down there. And then, of course, the Earthwatch Institute, which is where I got my start with primates, continues to fund students to go to field sites and work on scientific projects. So they have two fellowships of relevance. So they have one targeting teachers and female-identified high school age students. So if you're a woman hoping to get field experience, female-identified in some way, apply. And they also have one for LA area high school students in particular to go down and participate in these scientific field projects. So even though it can be financially very intimidating to do this kind of work, increasingly there are ways for us to kind of get around this so that folks who maybe think this is outside of their potential, you can actually go and get these experiences.
Yeah, and I was going to say the Leakey Foundation has a grant to the Joan Donner Field School grant or scholarship for students from countries with limited resources to attend field school. So, and we've shared links to, again, all of these different scholarships in the chat. So definitely check them out. Chris, thank you so much for joining us today. This was a really fun episode.
No, thank you. This has been really great for me too.
Thank you so much for watching this rebroadcast of Chris's episode of Lunch Break Science. We'll be returning to new live episodes of Lunch Break Science in July, so keep an eye out for more details soon. Now, I'll turn the floor back over to our rebroadcast for Chris and myself to sign off.
So thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Happy Pride, everyone. And until next time, stay hungry for knowledge.
Lunch Break Science is brought to you by the Leakey Foundation and is made possible by the generous support of the Ann and Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientist, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations will be matched by generous donors, meaning your impact will be doubled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.